Hello.
Hello, good morning.
So let's begin, please. Thank you, Lee. So good morning. Good morning, everyone. So today we have an in-house guest, our Dr. Armando Subastian, and he has a PhD in molecular genetics and microbiology by Pontificia Universidad Católica de Chile. So he has conducted many studies with microorganisms from the Atacama Desert. And he, among other things, seeks to understand adaptations to extreme desiccation while defining limits for life on Earth. And of course, its implications in astrobiology and biotechnology. So thank you very much for coming.
So today we're going to talk about our next expedition in which we are going to leave a, so to speak, safe environment of the desert and going into the sea. So I'm going to explain how do we reach to that point in particular. This started actually in 2009 when inspecting the coastal range of Atacama, in which by the characterizing the type of life, microbial life you could find inside caves, we found a particular type of microalgae that live under such conditions, which is a species of cyanidium. And cyanidium is intriguing because it seems that it's a very ancient microalgae. So ancient that when you inspect this particular species, the outer structure of the cell, it has this huge chloroplast that is actually has the organization, the typical organization of a cyanobacteria. So what we think in the case of a cyanidium is that it may be an ancient representative of the second endosomal basis of life, in which an ancient microorganism engulfs a photosynthetic microorganism that then turned to be the chloroplast. So we think that is in that stage for some reason and stay there. So to call a living fossil, so to speak. So that was intriguing to find why would you find such an ancient species in the coastal range of the driest desert on Earth. In another cave, we found more or less related finding in which not in the yeah. In which, yeah. So in which we found this microalgae in which we clearly found there was a new species in this case, another microalgae, as I said, of Dunaliella. And Dunaliella, this is the first ever found member of an entire genus that are all aquatic. This one we found it growing on a cable. Okay. That along another finding in which is 100 kilometers south, we found that under quartz stones that you find around, you will find these hypolytic biofilm that are comprised by at least 80 species representing the 3 domains of life. From there, we isolated a particular cyanobacteria in this case, which in the process of identifying what genus it belonged to, we found that in many cases, I mean, in most cases, you would just need particular genes that we know that are very informative to tell you what species you're working with. But in this case, it seemed that the genes or these particular genes were not enough, and we have to, just by that reason, went to look for other genes in order to identify which species was, in this case, one involved in the photosystem 2, since this is a photosynthetic species, of course, in particular, the gene that is playing the protein D1, which is involved in the photosystem Duna, has a critical role in photooxidation. And by sequencing that gene in order to identify it, we found that we could find all the 4 canonical members of the typical isoform of this particular enzyme 1,2,3,4 that are not well known for most of the bacteria, but we also found a very diverging one that was more related to the one of genes that we could find in cyanophages, that are the kind of viruses that infect cyanobacteria, that are in the process of moving genes and in and around a particular cell. It seemed that took out a part or an entire gene that indicate the PSBA. And also when we sequence and then we compare with that, we also knew that in this case, we have the molecular evidence that this virus at least was telling us that this species also was coming from the sea. So we have a number of findings, and actually we were able to see that particular virus when we look for it, making clear that that was the case. That along the finding that we knew later in time that all microbial life that is able to colonize the entire Atacama comes from the sea, driven by the wind that are throwing particles of dust, right, that are bringing all those life around into the Atacama. We knew that there was a number of findings that everything was coming from the sea. So that was the first step to start thinking, well, what is going on here? Why were we finding so many species that seems to be from ancient aquatic environment now they're colonizing the Atacama, and also some of them seems to be what we call, not properly, but that's the term we use, living fossils. Right, so all this, of course, have been published, so you can relate to all those findings in particular. And this one I like it because this is the first time I propose the evolutionary mechanism on how this is what's happening, which is a term that Stephen Jay Gould invented already more than 20 years ago, in which he proposed the term exaptation, in which life, by adapting to a different number of environment, does the metabolic route, does the genes involved, just by chance, that give you an advantage to another type of conditions. And that's the way you can adapt to new environments, okay? So as I said, this was in particular this publication, and we knew then that we have something was intriguingly going here. Why I'm finding species that are jumping from the sea to the desert, not only any desert, but the driest and all this on Earth, and some of them very ancient. So the question that followed, well, knowing that Atacama is also a very ancient desert, it has been more or less located in the same latitude for the last 150 million years. So explaining this climatic stability, that's how life perceive that climatic stability. So I start asking the question, if I have that, I mean, if I know this, the exposed portion of the Atacama is 150 million years old, it follows that the submerged part that goes into the ocean and meets the Nazca plate that is subduing below the South American plate, it must be as old, of course, 150 million years old. And if it is the case that the exposed part has this climatic stability in time, maybe the submerged part has it as well. So who knows what really old organisms, and in this case, it just goes from everything that we know in life, we may find here. So the question was, well, what else can I find in this onosis area? And the question was, well, how do I start looking for? I mean, looking for what that is really ancient, or just using that kind of terms, ancient living fossils, Atacama, trench, Nazca plate, and so on. We found that very intriguing question, very intriguing result, that you see all those points, those are all points that people just by chance, typically fishermen or people that were going for sediment course or anything, but looking for really ancient animals, found as I said, things that were not supposed to be still alive, like the Selacanthus, to give an example, that were only before known in the fossil record. So I give you some example here, and this is not just a 1, 2, 3 finding. There are many along, as I said here, as I showed here, or almost more than 6,000 kilometers. So this is not just a local phenomena. So this is all along the coast of Chile, because it makes sense that it's, again, you have again a similar environment. So I'm focusing here in the central part of Chile in this case, and the north part of Chile as well. Here, the area we're going to be studying. So for example, this one, Leptocheton, previously known only in the fossil record. People that found that they are living, well alive in the deep sea. Same thing with this one, this Sinisca. Same thing with the Neopilina. And actually, these guys, I knew it because I was born and raised in the Atacama. So I played with them in the beach when I was a kid. I didn't know that they were so ancient, and they were not. I mean, they were found as a species that were supposed to be extinct. Again, similar thing with the brachiopods, in this case, the Terebratulina, or Macandrevia, or Terebratella, or even, for example, in this case, diatom, for example, like Adonis. And actually, I have some of the fossils of this very same species, because at some point, Atacama was under the sea, the Nazca plate, pushing the South American plate, move everything up, creating the Andes Mountain, by the way. And so many of the species that people, even I myself, found these fossils, people found them living in the Pacific Ocean. So, well, this merit maybe an expedition to explore this place or what's happening there that explains why we have such a huge diversity of things that were known to be or supposed to be extinct. Okay. So this is actually formally started in 2014 when I approached Jean-Michel Cousteau Ocean Future Society. I wrote to the scientific director at that time. He said, oh, we are really interested in this. Let me just wait for Jean-Michel to come back because he was exploring somewhere in the world. And they even provided with a expression of interest letter in order to start looking for funding. With the bad luck that the poor guy, after 2 weeks after I contacted him, he died. So after that, I tried to contact someone else there, but with no luck. So that remained there for almost a decade. When in 2023, I'm sorry, there was a call by the Smith Ocean Institute, which is a non-profit organization based in the USA, which has one of the most advanced oceanographic chips you can find around. It is less than 1 year old, so you have the highest, the newest techniques for deep sea exploration, including rovers like this one that are able to go up to 4,500 kilometers down. Again, with the latest technology for sampling this type of environment, and this, I dare to send a proposal as a video. And they like it so much that they invited us to say the formal proposal written, right? And I said, well, we are really interested because we have roamed the world, but we have never encountered such a situation in an additional such an unexplored area. I mean, we are going into a truly unexplored area, finding with really good preliminary evidence, we're going to find things that you can find anywhere else around. I mean, for us, people are like studying other different type of deep sea ecosystem, you more or less expect what you're going to find. In this case, we're not actually sure. We think we're going to find very old things, but that remains to be seen. We have very good preliminary evidence that will be the case anyway. So what we're going to do, started in May of this year, we're going to start by the port of Antofagasta, which has all the infrastructure needed for the ship to arrive, for all of us, the team that are going to explain who is going to be doing what there. And we're going to go to actually the nearest sites, which for this kind of expedition are really close. I mean, 60-something kilometers for them is like going to the next corner, right? It's not like going into the middle of the Atlantic or the Pacific. So they were also happy for that it was something that it was really easy to get, okay? And with the advantage that at least we know the exact coordinate of previous findings. So we know we're not just a blind search, well, at least we know that we can go and visit the ecosystem that people found out. And so for me, it's like when you have these machines and when you put a coin and then you have this thing that grabs something like this, I mean, it cannot be a coincidence. For example, in this case, in just one sampling effort, you pick up 2 different very divergent animals, all the 2 of them in the category, oh, we have seen this as fossils. And again, and again, and again, it depends on how long you go along the coast of Chile. So what we're going to do is, there's a graph, the continental slope here. We're going to go up and down the slope there up to the limit of the rover, which again is 4,500 meters, which is a lot, 4.5 kilometers, that is a lot in the deep sea, right? Going there with the rover, and in a transit property, we're going to go up and down until we find the ecosystem we're expected to find, in which we know that those are just but a few representative of the entire ecosystem. They may be hiding there. So what we expect to find, more or less, is shown here, in which we have an ecosystem formed by different type of microorganisms, invertebrates of different kinds. The one I showed you, mollusks, and other similar things. Maybe also corals and other kind of organisms are also, if you think about it, if the ecosystem is really ancient, including the microorganism what we have been able to find, it also followed that maybe the microorganisms that are around and in them are also as old, right? So maybe the entire ecosystem, from the macro to the microorganism, is really ancient. So that's what we're going to target, find those type of ecosystem. We suspect that we may be dependent on cold seeps, because that is well known. The Nazca plate is bringing all this organic under the South American plate due to temperature and pressures, or those organics go up again at methane, up in there. So we expect to find something similar coming from below, something like this, right? That may be the source of energy for this kind of ecosystem, chemiolytotroph, so to speak, all that kind of life form that depends on microorganisms that are able to transform methane into a source of energy, then doesn't make it available for higher, so to speak, organisms that I'm showing here. So that's what we expect to find. But in this case, we cannot also forget that we may expect an important input of energy coming from above, because the entire area in front of the coast of Chile, given the Humboldt current coming from Antarctica, it brings so much nutrient that is one of the most productive areas of the world. Okay, and I have seen this in the area. For example, you can in anywhere around the coast of Chile, you can see zillions of birds that tell you that you have zillions of fish that can able to eat, and those fishes depend on the zillion of other microorganisms you can find on the sea. And actually, there is a particular parameter which is microbial upwelling on all the microorganisms that are from currents are coming from below and then going up and down in cycles like this. The highest value ever have been measured precisely in front of the port we're going. So you may have inputs of energy from below, from methane ships that we know that exist there, because people from the hydrocarbon industry have known, have sampled different areas along the coast of Chile, and they know that methane is being produced there, again, with evidence of how you put a plate under the other and all the processes that we know that happen, but also with given to processes like this. That remains to be seen on what is the main source of energy for this type of ecosystems. So let's start from the big things to the small things. In this case, Nicolaus Skissen from the University of Puerto Rico and Thomas Chile from the Florida Institute of Technology, people that has a lot of experience in this kind of expedition. They have been in deep sea submarines. They have taken samples with deep sea rover, etc., etc. They will be in charge of identifying all the things that we call whimsically called things with shells, which are the kind of thing I do not know, but they do. They know how to recognize such a species, again, by using traditional from morphology, but also molecular genetics, in particular genes that are very helpful for that purpose. Okay. Then comes Carlos Gonzalez from the University of Tarapacá in Chile, and Walter Sievel-Koval from the University of Arturo Prat in Iquique, both Chileans. They will focus on everything else that is not things with shell, like octopus, corals, and things like that, because again, in particular, Barter, he has like 4 decades of experience sampling those areas around, not that deep, but at least he has a well-known knowledge of all the fauna, the benthic fauna of the region. We follow in my case, in which I will be trying to understand what type of microorganism we find in the sediments, again, with the hope that we'll find truly ancient microorganisms. Then comes Felipe and Miguel. They will be able to, with a different approach on the sediment, tell us about the level of metabolic activity, the kind of metabolic routes, what they are doing there, in the et cetera, the biochemical cycles involved. Then follows Mara with her experience on viruses with the same approach. If we find really ancient microorganisms, it may hold really ancient viruses as well. So again, who knows what we're going to find in terms of really ancient viruses. Follows then Lauren Siller. She's from the Stockholm University. She will be focusing particularly in her area of expertise, which is the microorganisms of the methane ships. Then follows Peter Giggins from Harvard University. He will be trying to uncover the microbiome, that is all the microorganisms that are part of those things with shells, as I call them. That's his experience as well. Then Maria Sabel, he will start the dynamical entourage of all the system in order to understand, for example, how this ecosystem may be connected, how we explain nutrient cycling and all those processes that are related also to the geography of the place, which will be with a person from the organization itself, from the Smithsonian Institute, that are in this global problem of bathymetry in order to have the best maps available of wherever they're going. So knowing that, along other parameters we're going to be measuring, try to understand the connections in terms of fluid dynamics. Follows Daniel Carrizo, again, his expertise is on the kind of biosignature we can find around, because if you think about it, when you are inspecting deep sea ecosystem, you could start envisioning how would you be doing the same thing in the icy moons, like Enceladus or Europa, right? So having the experience of doing similar things in such environment can tell us on how, on what, on where exactly we should look for. Also related to that, Javier Sanchez, knowing that we have the methane ships and all the chemicals that are being produced and all the microorganisms that are involved, he will be studying all the process related to, for example, the biomineralization. Again, looking for putting that result in the terms of what would you expect to find in similar environment if you go abroad elsewhere in the solar system. Then follow Antonio. Antonio, he will be focusing the general geochemistry of the whole ecosystem, putting all members together on who is doing what in terms of the geochemical frame of on the entire ecosystem. Olga, in particular, will be focused again from the point of view, okay, so how do we understand this side? Again, if we go to Europe or Enceladus, he will focus on the carbon fluxes of methane ships, in particular, looking for on chemicals like this one, for example, clathrates. I'm not clathrate, and it's difficult to pronounce. In order to find what kind of such molecule we can find around, again, assets in the search of evidence of life. And this just started, and I think we're going to be also successful. We hope, and we're working hard on it, led by Olga and Antonio, in bring an in situ Raman spectrometer, which are the either those or two locations in the brown boxes there, in order to use a Raman spectrometry in situ to match. And again, thinking on how you were able to, and the technological hurdles on how to bring such instrument in a deep sea mission with the pressures involved, with the temperatures involved, et cetera, et cetera. And we're getting close to the last members. Jacek, given all his knowledge on the wide range of the microscopic techniques, from the most simple to the bright field to the most advanced, which is cryosem and all other experience he has, he will be involved in order to register all the whatever we find in terms of microscopy. And Andrew Palmer also, he will be focused in the general in ecosystem from the point of view that we must not forget that many important biotechnological applications have been derived on such environment. Let's just not forget that the polymerase that we use today, say, in our lab, was isolated from a deep sea microorganism, was found in a similar ecosystem like this one. So who knows what potential metabolite of interest that we may find here that we could find about interesting applications. He will be focusing on that. So again, all in the terms of describing a really new environment, but also on how we use such environment to try to understand how we approach the exploration of similar environment we expect to may be inhabited in other places of the solar system, like in this case of Enceladus and Europa. So I think it will be a really good, not only practical, but intellectual exercise on how do we design and think on how to actually do things with your own hands in an environment like this, where this is not, as you might imagine, it's not easy to get there, that deep in the ocean. So with that, I end, and I welcome that you are here, and please ask all the questions you want, and I'm happy to answer them. Thank you very much.